Good afternoon, everyone. My name is Joel Shinneman with the Office of Water Programs at Sacramento State, and I'll be talking to you today about a column study we performed on the PCB removal from urban stormwater using Biochar Amended Bioretention Soil Media, or BSM. So some background for those of you not in the stormwater industry. Bioretention Soil Media, or BSM, is an engineered soil consisting of 60 to 70% sand, with the remainder being compost. And this is commonly used in bioretention best management practices, such as rain gardens. And you can see in the bottom left is a cross section of a rain garden with the red circle around where the BSM is. And on the right, you can see a picture of a rain garden on Sacramento State campus during a rainstorm. A little background on PCBs. Polychlorinated biphenyls are a man-made chemical that was used in a variety of products until they were banned in 1979. The reason why it is still a pollutant of concern today, though they were banned from manufacture 40 years ago, is because they do not break down readily in the environment. They're a persistent pollutant, and they also bioaccumulate in organisms, which is why they're responsible for the current do not eat warning for surf perch and white croaker in the San Francisco Bay. So the goal of our study, again, is to identify a biochar media amendment that significantly improves the removal of PCBs by bioretention BMPs. So a little bit about our experimental setup. We use a column study. We had 6 columns, 5 used biochar amended BSM, and the control was a BSM only mix. The biochar amended BSMs are a mixture of 25% biochar and 75% BSM by volume. We used real stormwater collected in the East Bay area. We did this because previous sampling in that area let us know that they had elevated levels of PCBs. And for our study, we performed 4 tested sampling runs. Here's a picture of the column setup. We use glass columns with Teflon caps, and in them you can see the BSM biochar mixture. So jumping right into our sample results. Our first run, we have a nice high influent on the left. We can see a solid removal with our BSM on the far right. And in the center, in the green colors, are our biochar amended columns. You can see that most of them have slightly more removal than the BSM, one slightly higher. One thing to note is that when we performed this first sampling run, we saw that there was a lot of soil discharge from the effluent of our columns. So we performed 2 more runs that we didn't sample. We call them seasoning runs, where we ran real stormwater through them and just flushed a bunch of stormwater through until the effluent sort of stabilized. And after that happened, we did our second sampling run. And this is where you can really start to see a pattern in the results. Again, high effluent. Again, good removal by the BSM, the control on the right. But now you can see that the biochar columns all have a improved removal compared to the BSM. Then we move to our third sampling run. We had a lower influent on this one because we collected from 2 different locations. Again, solid removal by the BSM, and then we see slightly improved removal using the biochar columns. You see that there is a fairly high outlier in the rogue biochar. We were actually fortunate enough to capture a duplicate sample for this run, and the duplicate analysis results showed a sample concentration of 400, which would put it right in line with the other columns. So we think maybe something happened during the sampling run. Our fourth sampling run used diluted stormwater. We ran 1 part stormwater to 9 parts tap water, and the reason we did this was we wanted to see if a biochar amended BSM would be as useful in areas that didn't have elevated levels of PCBs, more of a standard level of PCBs for urban stormwater. So we got the influent much lower, and again, we see great removals by our biochar amended column. So in summary, the results of our column test show that biochar provides increased removal of PCBs from stormwater when added to BSM. This is a relatively small data set, so it certainly warrants further study. And that's it for my presentation. If you would like to know more, the full project report is available at the link at the bottom of the slide. And of course, if you have any questions, my email address is in the middle on the right there. Thank you so much.